Yeah, no, let me just introduce you. Hello, everybody. People are slowing entering the room. Today we have a special time. I'm not going to share any slides this time since you are used to already to attend the MTB seminar series. I would like to welcome you again. Um, just to remind you that the microphones would be deactivated, but if you need to make a question, you can either write in the Q&A area or raise your hand and I open the microphone to you. Um, we have stopped, we just finished the round of COVID-19 modeling research, and we are now open up for a broader aspect on mathematical biology. And the invited of this week, it's Anuj Mumbai. Anuj is an old colleague and a good friend. He's an associate director of Precision EO and instructional assistant professor at the Department of Mathematics, Illinois State University. Anuj currently also holds adjunct faculty positions at College of Health Solutions at Arizona State University and at the Precision Research Center, Berkeley. He is an applied and computational mathematical scientist whose research is driven by the quantitative modeling of problems of interest to the public health or social sciences communities. Anuj is a very active scientist. He has an extensive experience in the health decision analysis and in successful development of strong mathematical sciences training programs. Anuj is also a research fellow at the Center for Collaborative Studies in Mathematical Biology of the Intercollegiate Biomathematics Alliance in IBA. His research interests include the development of new cost-effective tools for improving the impact of infectious diseases while increasing our understanding of mechanisms that drive their prevalence and mitigate morbidity-related consequences. Anuj is currently working alongside with research of viral infection propagation, be through air travel, VIPRA, and global pervasive computational epidemiology, GPCE projects. Anuj, welcome. Thank you very much for joining us today. We are looking forward to hear from you, to learn from you. So please, you have the audience.
Thank you very much, Myra, and it's always pleasure to see you and your team, and thank you for inviting me for sharing my background, my research with everyone in your team and world over abroad. And I see many folks from different parts of the world. So greetings to everyone. Um, so my background is Arizona, where I am right now. It's Phoenix, Arizona. It's more desert land. Sun is rising. It's 7 past 3 minutes here in the morning. So greetings, everyone. And so let me share my screen. Um, so I was thinking about what to present. Actually, I think I am doing different types of work, COVID work also, and we just recently published some work which is now in press also, on COVID in air travel, during air travel. How can you understand it? What aspects of air travel really affect it? Um, but we are seeing so much of COVID everywhere. So I thought of presenting some different topics. Um, so today I will be presenting something on a tropical infectious disease, which is um, one of the key areas of research in my lab. Um, thank you, Myra, for giving the detailed background. And now I hope everyone knows. So I'm talking about community and community from infection perspective. Let me see, my slides are. So as we know, communities are not all equal, created equal because of various reasons. Have you ever met like anyone who's like you, truly like you? Well, first of all, what do you mean by truly like you? In what respect? So physically equal, mentally equal, ethically equal, financially equal. There's so many factors that makes it different from one another. Um, for example, how these viruses is spread in case of COVID, which is the latest news every day. We are hearing so many different things. they have different manifestation on patients, different types of patients. Some are severe, some are less severe, some are asymptomatic, some are pre-symptomatic, different varieties of symptoms. And so we are not all equal because of various reasons. Um, and there are different community environments that impact it in a multiple different way, and gives an individual varied risk for infections. So this is a very beautiful pictures that came in Medium. Medium is one of the news uh, on digital news agency, and it publishes very good articles. So today, what I will be talking about is complexity and scales in neglected tropical diseases. Now, I want to give this idea from a bigger perspective rather than mathematical model, which many of us already know about. How do you do the mathematical analysis? But I wanted to give you on a broader scale how we study these things. So complexity in a different scales. When we talk about complexity, the host vector, these are important populations in driving the infection. So what aspects of these are critical that makes it less or more epidemiological risk of infection? Spatial movement between the different regions, biting heterogeneities. These are all makes it very, very different, certain disease system. And so today I will be talking about a few examples from my research, Leishmaniasis, Chagas. Actually, instead of West Nile virus, I changed it at last minute. I will be talking about Lyme disease. Some examples from those end. So, the reason I kept this COVID and NTD, we have to understand we are thinking too much about COVID right now. And but NTD stands for neglected tropical disease, and these are people affected are extremely poor. They do not have adequate water access. And all the non-pharmaceutical public health interventions that are common for COVID, that the government are asking about to do for the COVID, to avoid COVID risk. Um, those things are not easily possible in populations which are affected by NTDs. So this is very critical, that we must fight them in tandem, and we can't just forget and only focus on NTD because, sorry, COVID, and not on NTDs, because they naturally have been a humongous toll. Is of course lots of people who are in these poor communities affected every year, but with the COVID, it has becoming more aggravated. So, for example, the co-infection with these diseases have kind of a different type of impact. It can aggravate the outcomes, but then its treatment can help also in COVID, because many of these COVID treatments have been NTD treatments, and NTD treatments has been for a long time. Peter Hotz is a very famous neglected tropical disease physician, global health person. He wrote this book, Forgotten People, Forgotten Disease, very interesting book, which is used in public health departments for teaching a public health course. Um, and he refers to many of these diseases are preventable, but not much attentions are being paid to these. In Chagas, for example, if you are infected with Chagas, this is a long-term disease. This is a lifelong disease. But all of a sudden, a person has a heart attack without showing much symptoms. So COVID can reactivate the infections. These pathogens are there in the body. So there are many issues with the co-infection that makes it focus on this diseases simultaneously. So, okay, so when I'm talking about neglected tropical disease, the first thing comes to mind is the vector-borne disease, and what is the parts that is impacted. I mean, if you see these colored map, um, It is showing the mortality of the vector-borne disease. Vector-borne diseases are much of the neglected tropical disease are vector-borne diseases. So the darker the region, the higher the mortality uh, in those regions. So it is on a tropical belt, which is the most and most resource-limited countries are primarily affected. Diseases such as leishmaniasis, Chagas disease, dengue, lymphatic filariasis, these are all considered as neglected tropical disease, but West Nile and Lyme disease are also vector-borne disease, not neglected part. There are different pathogens involved, species that is critical. There's vector species involved, and some are anthropootic and some are zoonotic. What that mean is that some are mainly in the humans are the primary reservoirs, whereas some others are other mammals or even birds and reptiles could be the host of that disease, apart from human being maybe an accidental host. So when we talk about NTDs in 2012, WHO, along with certain 10 big pharmaceutical companies, started to put money and focus on this disease. Before 2012, there was not much attention to these diseases. So it's a very recent way when we are started, although these diseases are, many of these diseases are more than 100 years old, and 100 years that we recognize, but it might be long before that. So so it is called the word neglected because it affects poor. It has pharmaceutical industry less focus on these ones, um, and basically the places where it is there have a weak infrastructure. So so in 2012 there was a London Declaration where all these WHO and these 10 companies, as well as Bill and Melinda Gates Foundation, which provided a lot of support, came together to decide on what are the diseases that would be considered as um, neglected tropical disease. So there were 20 diseases, 20 to 22 diseases that they finally said that will fall under this category. So they are primarily divided into viruses, protozoa, bacteria, and helminths. Helminths are the worm diseases, by the way. And from a modeling perspective, these could be subdivided into microparasite and macroparasite. They are modeled in a very different way when we write down the equations. So now they are talking about so in 2012 they had the goal of achieving certain targets by 2020, and I think only 50% of the targets were achieved. So, in spite of enough push from WHO, Bill and Melinda Gates Foundation, and many pharmaceutical. So, 2000 now they have come out with the next decade of global roadmap to end many of these diseases or eradicate locally these diseases. So this roadmap just came out. Note this is almost 1 billion. The places which are affected by these diseases are in resource limited countries, almost more than 1 billion people infected by these diseases. Now, when we talk about this, we know these things. One of the questions that comes to the modeler is what is important in modeling? And so whether it is domestic animals, mammals, or whether it is which insects, or it is a peri-domestic, or it is a wild sylvatic cycles, which aspects of those things becomes very important. So environments, modified environment, natural environment, or urban environments that could change the site. And the important thing here is the diversity in the pathogen, different types of insect species, different types of host that are present, and vector species. That makes it very different for this to identify what would be the appropriate scale in which we should model it. So I just want to give very interesting counterintuitive examples of NTD control that was seen in the past. So there's a disease called as visceral leishmaniasis. It is spread by sand flies. and dogs are the primary reservoirs. And so humans are secondary reservoir, not the primary one. Dogs are. So in Brazil in early 90s, they decided, well, if dogs are the primary reservoir, why don't we kill all the dogs? These dogs are domestic street dogs. So they started killing these dogs because their idea was if they kill these primary reservoir, there won't be infection that many. The prevalence level will come down. But but what they found is after I mean, these culling programs for the dogs from 1990 to 97, they found actually it has not brought down the number of cases in humans. Well, why not? In fact, the number of human cases increases. Why did it increases? Well, it increases because now, because of culling, you are removing the biting source. Now these bites has to go somewhere. They are hungry. Not hungry. They are needed for egg development. So basically, they were now biting more humans because those killed dogs were not in the cycle of this infection. So these interventions needs to be very carefully thought of. If you just randomly do some of those things, it may counterintuitively impact the disease outcomes in the population, human population. Well, there was another study in 2016. It's a very recent study from the state of Orissa in India. They found that certain mosquito species that are local to that regions rest in cattle sheds and feed on both cows and humans. They are zoophilic in nature. That means they are very close to human dwellings, cattle sheds, but they are feeding on kettles as well. Well, that means what that mean is, so bed nets and indoor insecticide spraying in human houses are less effective because they are not living. These flies are not living in those areas, and you are wasting the resources. So conventional tools doesn't work, and they just realized it recently. So what they did was um, well, they started to build these temporary kettle shed in the evening. They moved their kettle shed around the village. And what happened because of that, the the sandflies, which are responsible for this disease, sorry, the mosquitoes, this which are responsible for the malaria, are now flying in the periphery of the village, and that dramatically in the evening, and that dramatically reduces the number of bites on the humans. So they made these small kettle sheds around the village and moved their kettles in the evening. So that was a very interesting thing, that if you reduce the certain number of hosts somehow from the population, it could change the interventions and could bring down the number of cases in the environment. So very beautiful examples from India actually for the malaria. Another disease is visceral leishmaniasis. This is an interesting example. Here, this is visceral leishmaniasis is a very life-threatening disease. People die from it. It is spread by sandflies and not by mosquito, though. In fact, this is the second largest parasitic disease killer after malaria. Malaria is also parasitic. Um, so, but there is also a sequel of disease. Some cases of visceral leishmaniasis get post-Kala-azar dermal leishmaniasis, PKDL. But there is a difference between the two. The PKDL cases do not die from the disease, whereas leishmaniasis people die from the disease. So it's a mild infection, and only few percent, 1 to 5% treated for visceral leishmaniasis get. So it's a sequence of disease in only handful of and they can act as a same parasite, same, but in some cases they become a PKDL, whereas some other doesn't. Well, in 1990, they sprayed a lot DDT spraying to control this malaria, and that controlling the malaria helped it, mosquitoes for malaria, helped it in DDT is a insecticide. It helps in reducing the sandflies quite drastically. In fact, they found that there is in 1990s there was no infection. If you see, there is no infection they found. Well, the cases they did not take care of the PKDL cases at that time, and so in 1991 there was a big epidemic, and so they realized what they missed. So they analyzed the data. When they analyzed it, they found that the PKDL cases, there were very few of them that ignited this um, is outbreak. So what this example is showing is the host doesn't mean kettles or dogs, but it also means human, different types of human host, right? So here there are two sequel diseases, and one disease they were focusing on, which is most people get it. Only few fraction in the sequence get a PKDL, and they did not focus on PKDL. And so so the host could be different stages of infections as well. Very good example of that. Well, so these three examples are showing that hosts are very critical, different types of host, whether it is mammal, whether it is um, infection stages within the humans. This is very important. So a couple of decades ago, there was a term coined as One Health approach. Basically, that is taking into account that if we are focusing on public health of humans only in our mathematical models or in general policies decision making, we are missing the whole chunk. We have to take into account socio-ecological environment in which animals and veterinary approach also comes into place. They called it as One Health approach. Very good example of One Health is malaria, human malaria, and it has very contradictory impact. And some places, as you seen in the previous example, the livestock which are near to the humans dwellings can have a huge impact in the prevalence. So the kettles are basically some of the species of the mosquitoes stays in close to the livestock. Well, it helps more animal you will keep. It helps in diverting the bites, which could be many, could be infective bites from humans to animals. So that's a good thing. That's a benefit. But then if they are feeding more, if you put too many of kettles or too many of livestock and they are more biting on it, they are getting more feeding on it, then you are countering some other aspects, which is you are increasing the density of the mosquitoes. And if you are increasing the density, the infection can also rise because of increasing the density. So if you improve the blood source, it not only improves, I mean, it increases the vector survival and the density of it, though it has had a benefit of diverting the bites and reducing the infection in humans. So there's a balance you have to maintain, how much you should put the cattle or livestock close as an interventions. So relative contribution is very important, how much. So this needs a study actually. It's not that easy to say, okay, livestock, we can put many of those ones and we will solve the problem. But that problem has many end to it. So, well, what is critical factor relevance in vector-borne diseases transmission? So, well, it's a scale in which we are modeling about. Is it a spatial scale, temporal scale, quantitative scale, analytical scales? We as a mathematicians also think so. Um, what is an appropriate scales in which we should measure? That is something that we always focus on. And dimensions, whether the disease transmission route, how is the disease is getting transmitted. There are multiple ways the disease can be transmitted. For example, in Chagas disease, some mammals get this disease by mistakenly eating the bugs. Of course, by the bite of the insect that it is transmitted, but it is also spread by eating of the bugs by mistake by some of the mammals. Sometimes it is spread by mothers to baby, vertical transmission. So there's different ways transmission routes could happen. Okay. So the presence of different species, you were talking about, let's say, um, uh, host same, like let's say mouse. Sometimes there is in the infection cycle, mouse are important, certain, but there are many species of the mouse that might be present in that location. So species might be the presence, and the influences each one does may change the impact. And why is it important to study these scales and dimension? Is because it gives you where to put interventions. It's not like one size fit all you can do. So to make a greater impact, you need to know which scale you should put in scales or dimension you should put intervention. In mathematics, what we do is basically identification of threshold behavior. Like there is a book by Malcolm Gladwell, Tipping Point. It's a beautiful book. It has nothing to do with mathematics, by the way. But it simply talks about in real life, many times we see why certain things becomes a big problem, whereas certain other things doesn't become a big problem. So it is taking an example. This book is taking examples from real life examples. For example, Arab Spring. Have you heard of Arab Spring? It's like a mass revolt happens in the Middle East countries. One country started, Tunisia, it started by killing of one poor person, and it becomes a big revolution something like that in the whole Middle Eastern countries in early 2010. It started in 2010, and it toppled many government which were for many years they were there. But at the same time, there are these killing of some or small riots happen every place, and that doesn't become a news. So why is that happens? So Malcolm Gladwell talk about a real life scenarios and talk about why certain product become a big things, whereas certain other product doesn't become a big things in the market. So the very beautiful book, and accessible to everyone, not only mathematicians. Well, one thing we have to understand is why we are doing this work of mathematical modeling. I mean, it's not about prediction, although many of us are doing some of these things for the COVID, for example. I think prediction is a part of it, but not the primary part of it, I would say. It is must of, is talking about when can system tips or tipping point. So mathematical bifurcation translate well into epidemiological threshold, for epidemiological threshold like reproduction number. But there are other threshold like vector density, for example, vaccine efficacy. All are used as a threshold. Um, well, the same thing medical entomologist does it, and they call it as a vectorial capacity. They also do the similar things, but they don't have a sophisticated models as we have it. So their model is very simple, which is this VC is a vectorial capacity, which is what they measure in the field is number of infective bite received daily by a single host. How many bytes? Why is this important? Because that from that they can make it out how much infection is there in the population. Okay. Now the good thing in their model versus our model is, they take into account those parameters that they can truly measure in the field. There are certain things you cannot measure in the field because of various limitation. So they put those, and these parameters that you are seeing here are the parameters that goes in this formula. And that basically parameters, all of them you can measure. Okay, so their role is what we can measure, we will put it in. And their model is very simple. It's a function basically of various parameters. Well, this model is very beneficial. And if you look at carefully, the simple function is used for many diseases in a different way. For example, in leishmaniasis, humans are the primary reservoirs in certain part of the world. And so only treating humans is important there. Even though it is spread by the vectors, the treatment of humans are key here. And that comes from this formula. The yellow fever, killing the larva comes out to be better rather than a treatment of humans. Okay, so what aspects is there that needs to be reduced is M here in this thing, which is basically density of the vector per host. Okay, now in malaria, targeting intervention between vector and pathogens like reducing B here, that becomes very critical. Okay, so different disease you tackle in a very different way. You want to reduce this thing to reduce the vectorial capacity or number of infective bites. Well, there is certain limit. Of course, this is measured in the field, everything, but it is still very simple and the real life is very complex. So one thing that we all know about is a reproduction number, and it started with the Ross McDonald model for malaria in early 1900. And that is basically kind of a similar structure than this, but it has much more other features. For example, this gives us a bifurcation results, whether the disease will persist or not disease. That results you cannot draw from the medical entomologist formula, for example. So in this one, we can talk about it. Not only that, we can find it out what would be the infection level in human or infection level in vector in this mathematical model. So it is more sophisticated, but the structure is many of the structure. You don't want to put too many artificial parameter in the sense that you cannot measure in the field. Okay, so but the idea is similar, so that you can do much more over time, and at the same time capture lots of features that you can measure in the field. Well, there are major component of vector-borne disease. Again, as we are discussing, vector-borne diseases, the reason I'm talking about is a neglected tropical disease has many vector-borne diseases. So host, vector, pathogen, they all contribute to this. But what aspects of the host and what aspects of the pathogen or host or vector makes the disease system kind of a complete? Some are less relevant, some are more relevant depending on the disease system and the region that we are modeling. And the things that you put in the models describe the transmission cycle that we are focusing on. Okay, transmission cycle could be I've worked on Chagas disease um, in um, in wild host, and there are many cycles. There are many species of the vectors that transmit the same pathogen in different hosts, and sometimes you have multiple hosts, and one host has two vectors transmitting. So there is overlapping of the transmission cycle sometimes. Okay, so incidence for these type of disease becomes very important. Typically, we model incidence as contact rate times probability times number of susceptibles times the chances that you can find the infection or prevalence, I over N. This is a typical standard incidence rate that is given in many of our models. Well, if you take the contact rate as a constant, then you get something like we call it as a standard incidence. And this is what primarily used for human diseases. Okay. Well, if the contact rates are linearly dependent on population size, then we get it something called as a mass action, and that is used for zoonotic diseases, animal diseases. These are also called as a density dependent this thing. Now, why I'm talking about this is very simple for many of our folks. The reason I'm talking about it because this idea I'm taking it more complexly. So remember, this contact rate is constant here, and contact rate is linearly depending on population size. Well, in vector-borne disease, it is more tricky to model those things. So transmission terms are more complex. And let's understand how we describe new infection vector or transmission rate from vectors to infected host, to vector from infected host. So well, this first term is here is a maximum possible number of bytes per day made by a single vector. This is a biting rate, we call it. Then you multiply it by susceptible vectors. So that is the product of the first two is the total bytes by all susceptible vectors. Um, and then this proportion of infected host is the next term. And if you multiply the first three terms, you get a number of bytes per day by all susceptible vectors that are on infected host, on a single infected host. And then the last one is a probability of successful transmission of F from host to vector given a byte. Not all bytes will result in the transmission, only few of them. So that probability is very critical to model it. So this is the part that the product makes sense from a vector-borne disease. And this is number of bytes per day that result in successful infection into the vectors. Well, the same thing you can model from infected vector to host, but this is a little different than the previous one. Here you have again the biting rate by a single vector, but then you want to know, because you are talking about on the host, therefore you have to count how many vectors are landing on per day on the single host. So that is the next term. And the product of these two is the number of bytes received by one host from all vectors landing in the host. Next term is the total number of susceptible hosts. And if you take the product of it, this will give you the number of bytes per day received by all susceptible hosts. Okay, this is a number of bytes per day. And how many some bytes get wasted. Why? Because if they are biting on a susceptible host, the infected host, then it is not transferred, right? So infected vector will transfer only to the susceptible host. So proportion of infected vectors you multiply, so that gives the number of infected bytes per day seen by all susceptible host. And if you multiply it by successful transmission probability, then that gives the new cases generated by a single host. So this is the term, and if you simplify, you see the NV will cancel out in the second term, and this NV cancel out, and you will get a term like that. Note one thing: the denominator is all host in both the terms, is all host. And that is because, and it's correct, there's no error to it, because the way we interpret it makes it all host in the denominator. Well, I wrote a book chapter I forget actually, it's 2017. Here I talked about how these transmission rate varied based on which place you are modeling. If the place has abundance of host or scarcity of host, you have to know those things, or too many vectors per host. That assumptions lead to a different transmission terms, and therefore the analysis will be very different. So I talked about these things in this chapter, and therefore the dynamics, I mean, we just can't use the same type of infection terms everywhere. It depends on what are we modeling, where are we modeling. These are very critical aspects. Okay, modeling host and vector species. So there are different types of host and vector species that makes it very critical from the transmission dynamics perspective. Well, vector has preference. They like good food, just like we like good food, and so every species has a different definition of good food. So different vector preference. They like different types of blood meals. Um, of course, it could be livestock versus humans. So one place is livestock is kettles are, for example, one host, and other could be humans, for example. So they might have a preference over one another. In fact, not only that, they might have a preference over infected and not infected in some circumstances, some diseases. So if you see in this infection term, the denominator is showing the preference. Alpha V is a preference. So they preferred more infected people. than susceptible people. It has one coefficient. So alpha V is a preference for infection. IH and EH are the infected host, whereas S is a susceptible host. So sometimes this happens in the disease. Okay. Host competence is very important. Once the host get the infections, how good it can harbor the infection. So a good competent host is able to receive pathogen vector easily. It can able to transmit easily, sustain therefore extended. These are all, and there are different types of host. Accidental host that just gets the infection but doesn't show, doesn't harm anyway. And there are a little bit difference in the primary host versus reservoir host. There are different subtle difference between the all of these ones. One can produce the virus inside the, one cannot produce the virus. One can show the symptoms, one cannot show the symptoms. So there are lots of differences in that. So, and the level of infection population changes based on vector preference, host competence, abundance of host and vector. These are all factors that changes the dynamics. Okay, so let's talk about some examples very quickly. Study one is we are talking about ecological community in the visceral leishmaniasis. This is a research that just got published. Actually, last year it got published. And this is talking about complexity inherent due to many structures in leishmaniasis and how that changes the threshold of elimination of the disease. So let's see what we talk about here. So in this one, excuse me. So there's a lot of reservoirs like kettles and like cows, goats in the India. In fact, there are when they took the blood meal from these dissection of the mosquitoes, we found that they have the blood meals from a different host, and this is the data from India. Um, so we built this model minimum. The question was to understand how we can in many different complexity, how we can understand the threshold behavior of elimination. So we constructed a minimal models, which was basically having multiple hosts, two reservoirs, one human host, so three total host population, and the sandfly population. So this is a caricature of it. And then we have multiple patches. And so we had a human movement also between the patches. Okay. Um, and each of these population in the patches were divided into human, sandfly, and reservoirs, the two reservoirs. And in the humans, um, we have post-Kala-Azar leishmaniasis, which is a sequels of the visceral leishmaniasis. So remember that we were talking about two diseases, PKDL and so there is a PKDL compartment K, and I is actually a visceral leishmaniasis. So there are many features of complexity that is seen in the visceral leishmaniasis that is there in this minimal model. The goal was not to make it everything in that. The goal was how these, because if you make it too complex, you cannot analyze it, right? So minimal model, how we can use it with multiple types of complexity and understand what is the role of each one of it. We consider biting heterogeneity. Not everyone is getting the same number of bites. Okay, so some kind of features that is seen in India, we captured it into this model. So for example, number of bites per individuals were Poisson distributed with the mean some things. And the mean bites were gamma distributed. And this is what we used practical information from the field to generate some of these distributions there. If you capture this thing with a little bit calculation, and the paper talks about the more complexity aspects, you can come out with an incidence term, which is not a typical incidence term. It has a log inside it, and it is modeled in a very different way. Okay. and the sum here, if you see there is a summation sign here in the denominator, this is over all host, and alpha j's, alpha L is actually the preference for each one of it, each of the host species. Now, we also incorporated short-term human in certain parts of India. People are day laborers. They go and work in certain places and come back in the evening. So short-term mobility. So we incorporated the residence time matrix into it. And this transmission term that you saw it in the previous, this thing, if you add a residence matrix, it becomes more complex, but again, in a simple way, feed capturing certain movement between the patches. And this is, if you carefully see this more transmission term, gives you kind of incidence rate of this form. Now, again, the details are in the idea is how you capture these things in a simple way in this model. So if you do the analysis of it, you find typically the standard is to find each patch reproduction numbers and how is this different parameters of vector preference, host competence comes into this picture, into this formula, which is there in this formula. But the point here is, main point here is, in vector-borne diseases, you do not really take R naught in analysis in threshold behavior. It is the threshold, the vector density become very important. Why vector density? Because that you can bring it down. That you can play by insecticide spraying or by certain types of vector control program. So R naught is not used as a bifurcation parameter many times. It is the vector density that is used as a as this. In fact, what we found here is sandfly density thresholds for eliminating this disease, leishmaniasis, is lower when we incorporate movement, spatial structure. So threshold changes. The threshold changes if you add the spatial complexity into it. In fact, it becomes lower. If you analyze this thing, there is a backward bifurcation, which is basically saying the typical R naught greater than 1, you can control the disease, and R naught less than 1, not control the disease. I mean, R naught less than 1, not control, and greater than 1, control, is not the standard way you can capture in this thing. Well, there is a region that is, I mean, you need to reduce the disease less than this level in order for it to control the disease. So there's a threshold level. And if there is initial condition of your model, or if your local region initial number of cases is above this, then you could still have disease in the population, even if R naught is less than 1. Okay, so that is what the structure that we are seeing in this models. There's a bistable region, which is, and this bistable regions is a function of disease death and also the movement rate. So, and as the disease death decreases, this backward bifurcation becomes a forward bifurcation. Again, this is also a function of movement rate. So if the movement between the patches, you play with that, these bifurcation would change drastically as well. Well, in fact, what we find is the biting heterogeneity, the variation in the biting in the population. So some, they are biting more than others. That increases the sandfly density. Again, this is a sandfly density that we are using as a bifurcation parameter and not as an R naught. This increases the threshold. So the threshold is increasing, and the prevalence is decreasing as we are increasing the heterogeneity. That means the variance in the biting is increased. This would be, okay, so I think I have a, okay. Okay, so next is, well, there's many results in this work. And today I don't want to bore you guys with many results, but rather show you the big picture of it. We also talk about integral stability, which is basically linked with the basin of attraction in the bistable region. It is very important because it gives you how resilient your system is in a multiple threshold, a multiple equilibrium region. And basically it can tell you if under different scenario, for example, we took many models into it. In one model, the host was dead end host. Like the animals were dead end host. In the other model we took, the animals were secondary host that were good competent of the infection. So we took many different scenarios there, and we looked into this resilience of the system. And it is found when there is a dead end host, disease-free equilibrium is much more resilient in the system. So little bit perturbation in the parameters, which are epidemiological parameters, will still remain you close, good towards the disease extinction, elimination. This is very important for an intervention from elimination perspective. And so it gives a very good picture, even in spite of this complexity inherent in these models. So integral stability is used as a measure of resilience in many of these things. Second study is talking about modeling the impact of domestic host in dynamics of cutaneous leishmaniasis. Well, this study is in Ecuador, and we did it. I don't even remember when was that. I think it was in 2019. Okay, published, but we did it in 2016 when we started. And there, here we were focusing on this one particular province there, Santo Domingo, and basically we wanted to know is what are the other hosts there. So nobody ever has measured the host in many parts of. I mean, the vector surveillance is very weak there. So we went in the field actually and measured it, and we found that the vector species are feeding more on domestic birds like chicken, basically, than mammals. They are feeding more. But okay, so that's a good thing that we found from the data, but that is not sufficient information. So then we build a models to understand how good are these birds, the chickens, in transmitting the infection. So what we found, even though they are biting preferences towards the birds, the vector sandflies, but the birds are not good transmitter of infection. They are not transmitting it back there as compared to humans. So that was the basic study, which was a combination of empirical works and the mathematical models. Very interesting work, and quite a good result actually in there. The third study, and I think I will end very quickly. This is, I think I'm in the last stage of my few slides. So tick-borne diseases, Lyme disease is very common in US and actually in many parts of the world. But in northeastern US, it's very common. And it is very complex cycle of this stages that and the host that kind of led to this transmission of infection. So these ticks, which are the vector here, are their lifespan is 2 years. They have different stages, and different stages happens in different year of the seasons of the in the year. And it can feed on mammals, birds, many of these things. And they basically is a very weird way. When these mammals or host walk in the grass, they kind of tag, they are in the grass, and they tag around on the body of the host, and then they bite and suck and they transfer the infection. So what we were looking at the role of bait box vaccination program in the mouse, because mouse is considered as the main reservoir host. And well, we all so what we found there is that it can eliminate the disease. The vaccination rates, if you could do it reasonable enough, which is achievable, because we are talking about wild here. It's not easy to find a mouse there in the wild, all of them, in fact. So if you cross a certain level of vaccination, of these mouse, and vaccination is done in a very special way by baiting, by putting it bait, and then they get attached to the body somehow. You can eliminate this disease, but it depends on duration of vaccination. You can't just do it one time, and this, it depends on a season, which season you do it. So very important results from this disease, work. And it shows vaccination is, what it is doing is basically it is switching off certain population of the humans from the transmission cycle. Sorry, the mouse from the transmission cycle. And therefore you are having two types of host now, vaccinated and non-vaccinated. So the transmission cycle will be disturbed if you change the number of vaccinated in a certain way. So very good results, by the way. This fourth study is between Madrid, Spain, and Tolima, Colombia. We studied 2010 outbreak in both the places. And Here we took very different regions. Colombia, which is a developing country, versus Spain, which is Madrid, especially, is a very developed city. And in both cases, we analyzed the data. We found in Spain, it is the domestic dog which is transmitting the infection, whereas in Colombia it was different thing. It was, we found that it was a movement of the military personnel from the wildlife and coming to the urban areas were causing the outbreak. So very different way the disease was. Both are urban areas we were studying, but the mechanisms in which they were spreading is very different. um, Madrid outbreak was very severe than Tolima. Again, Madrid is a very urban city, very good economically, and in spite of that, it has a larger outbreak than Tolima in Colombia. And the reason was the disparity in the health infrastructures. There's lots of extreme poor and lots of very rich in the Madrid regions. Um, so there were lots of distinctions in these things, and what we were showing is the mechanisms of these complexities very different, even though the same disease we are talking about, but different districts, different cities. Sorry. So I'll end here. Thank you very much for listening to me. I think I have taken a lot of time, and I want to have few minutes for question and answer, if there is any.
Thank you very much, Anush. It was very interesting to get an overview of all the work you have done in vector-borne diseases. We should catch up on that actually, since back in 2000 and blah, guess 12 or 13, where we met last in Nimbos.
Yeah,
We started a very nice leishmaniasis model, and I never had the opportunity to continue that, but that would be very interesting.
sure.
So for the audience, if you want to make a question, please raise your hand or write in the Q&A. I normally start with the question, so I give time to the participants to think and to digest all the information. So I have many. I will not ask them all, but some of those are of my main interest. Um, what you called minimalistic model for the leishmaniasis with patches and etc. It looks to me very complicated already, and I like complicated models, you know. And I was surprised that you found this backward bifurcation. Is there any other parameter scenario that you can find a more complex dynamics rather than only a backward bifurcation, even though it's may probably not biological, but
Yeah, so if you see the transmission term, there was actually having a logarithmic term, because we took the biting heterogeneity as a distribution, and we approximated certain ways to get this final simple. Actually, logarithmic term was very simple in term. So it was difficult to do a comprehensive analysis as of that thing. So we took. For example, we said it is a dead hand host. So we kind of switch it off the transmission from certain host, certain reservoir host, and analyze
mhm,
the system in a very simple way. So the model was, we were unable to analyze the complete model, but it was analyzed in pieces in a
mhm,
certain scenarios. And the one that we did it, we could able to do the local behavior. Again, the global aspects might be interesting to look at. It is possible that if you put these complexity into it, there might be kind of hysteresis of some sort, or periodic oscillations. It is possibility there, but we are not claiming that. The goal there was, how do we describe the threshold on these parts in a few things into take in account? Because if we wish to take all these aspects, like we take biting heterogeneity, we take movement of the people, we took reservoirs, competence, vector preference, all of these things are there. But again, if you can't do rigorously the analysis, then it is a waste. So we took combinations of those ones rather than all at the same time, to come out with a reasonable understanding about the threshold behavior, which was a local behavior.
Mm-hmm.
And other things we did it simulation based, to make sense out of the system. So it's a combination of many of those things. The goal there was how these things come into play, like biting heterogeneities come into play when we talk about threshold behavior.
Mm-hmm. Well, that brings me a second question that, I mean, we are now all talking about COVID and we need data to validate the models and so on. When we talk about vector-borne diseases, data is a very sensitive subject, because although we might have data on the cases, it depends how people notify them. But I mean, it's basically impossible to get good data on vector.
Mhm.
So my question is, how do you validate or you parameterize your model, knowing the difficulties on availability of data, especially in vector
Yeah.
data?
Well, that's a good point, and I think there is no one answer to it. Again, the what actually I have seen, Myra, your work also. We work on theoretical understanding of the system. So I think understanding what goes in the system and what is the impact of different mechanisms is very
Yeah,
key
yeah,
before we even talk about prediction of some
yeah,
sort.
I
And
totally
that
agree.
is what your research lab and my work has been actually primarily focusing on. And this is, as I was seeing in these counter examples that I gave, I mean, people in Brazil started killing dogs for like almost a decade, right? Domestic dog.
Yeah,
And that resulted in a counter infection, you know. And there's no need for doing this thing if we understand the mechanism, how we drive the system, right? So that 10 years of killing of street dogs was unnecessarily, and the cases were well, didn't even increase the, it actually counter backfired this is policies. And so our work
yeah,
has led to understanding of the system and properly focusing on interventions in the right place. Now we are not telling how much of intervention we should put, because we have not used
yeah.
the practical data, right? But we are still telling you, instead of randomly going there, you should focus on these aspects of the mechanisms.
Yeah, but still, we have kind of an idea of the average where the parameters should go. So we cannot
Right.
be completely out of,
That's true.
natural
That's, yeah.
parameterization.
Yeah, that
Yeah.
is true.
Well,
And
it looks
actually,
very interesting.
go ahead,
Yeah.
Matt. Yeah.
No, no, sorry, I'm overlapping you because there is a kind of a delay. Maybe
Oh,
in the
okay. Yeah.
afternoon here it's
Yeah.
terrible. So I let you finish, and then we would have a question for one of the participants. Anush, do you want to finish your answer to me, or sorry that I
Well,
interrupted
it's
you?
fine. I think I've answered. Yeah. It's
Yeah.
fine. Yeah.
Okay, perfect. So Amit, you are able to ask and switch on your microphone. I have just allowed you to come in, please.
Yeah, first of all, thank you MTB Group for organizing these lectures. And about today's lecture, Professor Anuj, very interesting and interactive lecture with lots of real life examples, inspiration for beginners like me. And I always follow your posts. And maybe my question is irrelevant for this session. Actually, I wish to know what are your predictions for current position in India?
You mean to say COVID?
Yeah, yeah,
From
COVID. Yeah.
Amit, I am guessing that you are from you are in India right now, right?
Yes, sir. Yes, I'm in Punjab, India. Yeah.
Okay. Well, I hope all is going well. First of all, with you and your family and everyone in your family, extended family, as well as rest of the India. I know the situation has not been very good, especially what we are hearing in the news. Um, well, the prediction, I'm not work. See, there are different. I want to focus this thing very specifically today. Um, there are different set of modelers. There are modelers who focus on understanding the systems, and we as a mathematician do many of those things. Prediction is a very, very difficult task. Prediction is not a magic. It has to come from the data. The data is suppressed heavily in India due to many reasons, due to not having, I mean, the system is broken down in some places, some other issues, also political issues, and so on and so forth. I don't want to go more technical, but you see, it is very difficult to say in the lack of some information. If there is a delay that we know in the data, we could understand. But this is not about delay. There is many things that are happening that would, even with delay taking into account, um, the data is not really, for example, the hospitals are overwhelmed right now.
Yeah.
If you are overwhelmed, how will you count the number of cases? The only time the surveillance system, the way it works in India, is by counting people who comes to the hospital, right? So the more people that are getting infected, we have no idea to what level it is. And if you do not have an idea, you cannot do a good prediction. In fact, you can do any predictions, no matter what. And so that's one point. The second point is, some of the folks are doing work of prediction using one single model. You know, even we do modeling work for climate. It's a very sophisticated, big, huge models that we do it. And it's not a single model, it's an assemble of models. There's like thousands of models that we use, and we aggregate the results from those ones. And here, many of the predictions are based on single models, so which is not considered true in that regard. Third point, um, the prediction that climate model gives is mostly 7 days. That is a good prediction. You can't predict for 1 month. It's impossible. I mean, there's so many limitations. So I think the prediction is the only thing I would suggest, rather than believing on prediction, consider a prevention measure as the best option right now.
Yes.
And this is as a community advice rather than from a modeling. I don't have a very good answer to that, and I'm not modeling on the prediction as well. But these things makes me think whether we should take this modeling study results for prediction, give any importance to them, or kind of restrain my understanding on that.
Okay. 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 Thank you. Thank you very much, sir. And
Sure.
thank you very
Thank
much,
you.
sir, for your concern about me and my family. And I convey same for you, and pray to God. And I want to convey that I have already gone through this COVID infection, but the symptoms are mild. Now I'm okay. I have already
Yeah. Well,
gone.
Amit, let me stress that fact also. The prevention methods are very, very important right now. In spite of that, my family has also was infected, and now they are okay. But reinfection there is a possibility. Even with the vaccine, there is a possibility. Vaccines is good. You should take it. But there is a possibility. So that's things we need to keep into account, and a prevention methods needs to be rigorously followed, even if person has been infected before.
Yeah.
And
Yes. 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 This is needed. Actually, I got infected after the first shot.
oh,
I've
okay.
already taken the vaccine first shot, and after 10 days, I got infection.
I
But
see.
with the grace of God, now it's okay. But
Yeah.
but situation is not good because so many mutants are there. So I'm also worried about that. That the reinfection, that we have to worry about that. We don't have to think that once infected, then we have 3 months or 4 months to get reinfection. But due to lots of mutants, it's not sure that how much immunity we have and what type of immunity we have.
Yes. That's true. Thank you, Amit. That's a good point you made. Yeah.
Yeah, actually, sir, my question is regarding. I asked you this question because, as the world knows, that we are going through the second wave of COVID, and which is very horrible actually. And now the news and many predictions are coming that third wave is ready for third wave is coming also, and it's already more than 1 year that we are facing all this situation, and we feel that this 2021 is again a deadlock for us.
Yeah.
And we pray to God that this will end soon, so that we can meet physically and these events can happen offline also. Okay,
Sure.
thank you, sir. Thank you very much.
You're welcome.
Thank you, Amit. Thanks, Anush. We have a second question from Prasad. If you can open up your microphone, you can make your question, please.
Hi, Anush.
Hi, Prasad. How are you?
Hi, I'm fine, Anush. Thank you. I have a question on this vector-borne diseases. Most of these vector-borne diseases, can we control them? You have said that there is a choice of preference for what is that food, all these things. Like biting, whether they can bite humans or they can bite or can't we, this is a vector-borne diseases, can't we introduce some other species which can feed on these vectors and then control them, like some biological control?
Yeah, well, you made a very good point, Prasad. Prasad, I hope your family is doing well, by the way.
Yes, everyone is fine. Even I got vaccinated, so I'm very
nice.
No.
So you made a very good point. Of course, there are larval control. There is lots of biological aspects that is there. Now, in some of these things, Wolbachia, for example, is used to modified behavior of the vectors. So these are there are certain fishes are used to eat, and those are very good points. So, but the question that I was raising is today's discussion was, it's not like once you can do biological control for all the disease. Well, in dengue it might work, but in malaria it might not work. That's one could be. Or even in dengue, some region it could work and some other region it doesn't work. So we need to understand the biology of these diseases at the local level first. Before many times we are focusing more on mathematics rather than the cycles or transmission cycle aspects in that particular local region. And that is what, even though we are not predicting in that local region, still we need to understand the different mechanism that goes in a particular region. As I was giving an example of Orissa, these cattle sheds were responsible for major infection, and just moving the cattle shed in the periphery of the village, they were able to control drastically the infection level in humans of the so there are intervention you can design in that regard,
like reservoir buildings and reservoir buildings.
Right. So changing in the density of certain reservoir, that is primarily causing the cycles to dominate. So shifting the patterns, but you can't just shift anything. You first need to understand which things we will shift that will make a huge difference.
Okay. Thank you.
Thank you, Prasad.
Okay. So are there any more questions or comments or suggestions to Anush from the participants, from the panelists? Damien? Well, if not, I would like to thank you again, Anush. Thank you very much for taking part of our MTB seminar series. It was great to see you again and to learn from you. And I'm looking forward for the moment we can meet again in person and to continue our collaboration. Thank you, everyone. And I remind you that the next seminar would be on May 21st. Again at 4 o'clock, because our invited speaker is also based in the US. Thank you.
Thank you, Mara. Thank you. It was amazing.
It was great. Thanks a lot, Anush. It was a pleasure to receiving you and listen from you.
Same here. Same here. Take care.
Bye.
Bye.